Okay, so this is getting us ready to do station 1, which is DNA replication. So we're going to simulate what DNA replication is like. So the paper that you should have looks like this, except you have it in miniature version, right? It looks a little like this. And when we're done, you'll fold it in half and glue it into your notebook so you have it in your notebook for reference. Okay, so the first thing you're going to do is answer the first 2 questions about where this happens in prokaryotes and eukaryotes. And then you're asked to assemble the strand of DNA, and they give you the template strand, and they tell you what letters to use. So I'm going to show you a picture of these nucleotide bases that we're going to be adding together. Here's guanine, we've got cytosine is the blue, thymine is the yellow, and adenine is the red. So they tell us to assemble, and I'm going to use this big guy here. They tell us to assemble, so this is the template strand, or half of the strand, because remember, DNA is a double strand. So this is the one that they tell us to build. And through the magic of video, here is our fully assembled strand. So I'm going to go ahead and let you see from the beginning, CTAC, here we go. So I followed all those letters all the way to the end. Okay, now the next thing that they ask us to do is build the non-template strand. So do you see how in our template strand all of my arrows are pointing to the left? Now here comes our non-template strand, and remember it's complementary, so I'm going to scooch this up so you can see it. And ta-da, here is our complementary strand. Now do you notice, right, do you notice that it's T's that are fitting with A's, C's that are fitting with G's, and here it goes all the way down the line. Okay, so here's our completed strand of DNA. So something also I want you to notice is that in our template strand, the arrows are pointing to the left, but notice in our non-template strand, the arrows are pointing to the right. Remember, we've looked at the fact that DNA, the strands are anti-parallel, right? They're going in opposite directions, but that is the way that they fit together. So now you'll want to go ahead and record what the non-template strand bases were that you saw that we put together. Remember these guys? So you're going to write, for example, A, T, G, C, and so on. Always complementary to the bases in the template strand. So let's see if we can get through this whole double strand of DNA so you can see the whole non-template strand. All right, so after that, they're going to ask you to answer a couple questions, like number 5 and 6. So I'd go ahead and pause and go ahead and answer those, and then we'll move on to number 7, which is beginning the process of DNA replication. All right, so I have this model here of helicase, and we're going to start meeting. So in our cells, there's an enzyme called helicase, and it comes to the DNA. So now we're going to take a look at what happens as DNA, right, helicase is going through the DNA. Do you see what the helicase is doing? Your paper asks you questions about that. Okay, so we're going to keep going. Okay, now this strand goes this way. And it has to do with the fact that DNA is only built in the 3 to 5 direction. Keep going, get to the active site. Okay, meanwhile, this strand is going to town. So let's add a few more. Meanwhile, on the bottom strand, helicase is unzipped it enough for this DNA polymerase to come back to here and build a few more nucleotides on there. Okay, so this is where we are so far Do you see, sometimes what this is called here at Helicase is called the replication fork, because do you see how the DNA kind of forks at this part? Although in reality, it's usually a bubble, so usually it goes into the middle of the strand. So this would be, all this area out here would actually be, opening and still actually a bubble. Remember, there's billions of base pairs of DNA that need to be copied. So usually Helicase starts lots of bubbles of replications so that it will replicate faster. Remember, you calculated how long it would take if it only did one replication bubble at a time. Now we're going to finish the replication of the leading strand. And we can finish up the making of the lagging strand. Okay, so we're done making these. Now what I want you to notice about these is we started with one double-stranded DNA, right, with colors on both strands, and now I have my new DNA strands. I have two new DNA strands. And here what I want you to notice is, remember when we replicated this, we used gray for the new ones, right, and the original ones were colors. So do you see how half of my new strands now are gray and the other half are colored? And this comes back to the idea of semi-conservative replication, meaning semi, right, being half, and conservative, meaning I'm conserving or I'm keeping half of my original strand, and I built on the other half with new nucleotides.